the bacteriophage along with its DNA got only some specific genes or restricted genes, especially those genes which are present near to the prophage DNA here. If it gets only those genes or restricted genes, then this is called specialized transduction, and it is also called restricted transduction because restricted number of specific number of genes are transferred to through these bacteriophages through other bacterial cell. So this transduction is called specialized transduction. And this bacteriophage in Escherichia coli, the lambda bacteriophage has been specifically studied for this specialized type of transduction. So we can differentiate between these two transductions: generalized transduction, in which the whole DNA of the bacterial cell is converted into fragments, and any of these fragments can be transferred through bacteriophage to the other bacterial cell. But in case of specialized transductions, only specific parts of the bacterial DNA which are present near to the prophage DNA, they are fragmented, or they have been partitioned, and they are entered into the bacteriophage structure and can be transferred to the other cell. Not all the bacterial DNA is able to be transferred to any other bacterial cell. So this case is called specialized transduction. So we can differentiate between two of these, the generalized transduction and specialized transduction. Here you can see, here is generalized transduction in which all the DNA of the bacterial cell is fragmenting, or when it is fragmented, it becomes a part of the bacteriophage structure when these bacteriophages released by the lysis. Some of these bacteriophages consisting of normal viral DNA, their own DNA of their parent bacteriophage, or some of these contain the bacterial DNA. And these bacterial DNA bacteriophage, when they will infect any other bacteria, they will cause transduction, but not these bacteriophages which consist of only viral DNA. So during these generalized transduction, some of these bacteriophages which will contain the fragment of bacterial DNA, they will cause the transduction.